Food allergens are a tremendous concern for consumers and for the food industry, particularly when they are not food ingredients but contaminants, entering accidentally the food supply chain because of cross-contamination due, for example, to agricultural commingling and carryover when storing, handling and packaging raw materials and food products. The unintended presence of allergen can cause food product recalls as well as, in some cases, food allergy incidents. Food allergen labeling for food ingredients varies across the world, not only in terms of number of priority allergenic foods, but also in terms of types of food ingredients that are listed in the regulation. An outcome of the recent FAO-WHO expert consultation on risk assessment of food allergens is the review of the priority allergenic foods across the world based on data on prevalence, severity, and potency. Food business operator, when managing an intended allergen, should use an evidence-based approach, starting with a qualitative risk assessment based on hazard analysis and critical control points to evaluate the likelihood of cross-contact and contamination over the entire production process. This should be then followed by a quantitative risk assessment supported by reference doses. These reference doses are extrapolated from clinical data and are expressed as milligram of total protein from the allergenic food ingredient that is considered being safe for the allergic consumers. The reference doses are then translated to action levels, which is the concentration of an intended allergen above which action is required, by taking into consideration the average amount of food that is consumed. The action level defines the analytical measurand. So it is clear that accurate and reliable analyses are required to support the quantitative risk assessment. And the key performance criteria for any method used for specific foods should have a limit of quantification at least 3 times lower than the action level that has been determined for that food. So how to measure allergens in the food industry? There is no direct method that can quantify total allergenic food proteins, so we need to use markers. First-line methods are PCR and ELISA. PCR indirectly measures DNA as the target, while ELISA makes use of antibodies to quantify allergenic proteins. Both methods are rapid, simple, and economic, but they actually have limitations. PCR is useful when analyzing raw ingredients, but suffers from matrix effect, and the DNA also degrades during food processing. Its presence does not imply the presence of the allergenic protein, and it is also challenging to convert the number of DNA copies to the measurand. The ELISA measures the hazard. However, conflicting results may arise from using different test kits depending on several factors, and the results can also be influenced by the food matrix and the food processing. Isotopic dilution mass spectrometry is used as a confirmatory method that can provide allergen absolute quantification with a small measurement uncertainty by removing instrumental bias. It makes use of peptide markers as surrogate of the parent allergenic protein. This approach requires a deep understanding of the food system and the analytical pipeline. It is a step forward, making allergen analysis metrologically traceable. All these detection methods can either measure single peptides or proteins, while the allergenic consumer protection and the action levels to support the quantitative risk assessments are based on the total mass of the allergenic food protein. Therefore, it has been agreed by the food allergen testing community to express the analytical results as milligram of total protein from the allergenic food per kilogram of food. In the effort of harmonizing the reporting units of different analytical methods, the use of reference materials for food allergen analysis is crucial, and this action would make measurement meaningful for the risk assessment community and also comparable worldwide. Sample preparation process for isotopic dilution mass spectrometry analysis is crucial and follows a bottom-up proteomic approach, where proteins are extracted from the food matrix and digested with enzymes in order to release the peptides. These peptides are chromatographically separated and analyzed by mass spectrometry. And the answer we get is a ratio measurement of the natural peptide versus its isotopic label form. An external calibration applies to translate then the peptide ratio measurement into a concentration. We then need conversion factors to translate the concentration of a single protein in milligram of total protein from the allergenic food per kilogram of food. Different factors can influence the quantification of the peptide along the analytical pipeline. First is the selection of strong and reliable peptide markers reporting for the allergenic food protein. These can be either selected from literature or in silico, and they need to follow strict criteria that aim at enhancing the confidence of the marker. Candidates should then be validated in the food matrix by discovery mass spectrometry to confirm their presence and to assess the extent of modification that could have been induced by the food processing. Also, the extraction condition can strongly influence the mass spectrometry peptide response and may cause inaccurate results. Food processing can change protein solubility, and this may require dedicated extraction approaches that could be allergen-driven or even food matrix-driven. A poor method development could lead to poor quality results and therefore incorrect interpretation by risk assessors. So accurate measurements require a deep understanding of protein behavior in the food matrix. The accurate quantification relies on the equimolar release of the peptides during digestion from the parent protein. And this should also be validated experimentally, since some peptides may not be stable throughout the analytical pipeline, leading to underestimation. So isotopic dilution mass spectrometry can quantify accurately the peptides. We then need conversion factors to report the value as total allergenic protein mass fraction. With well-characterized food ingredients, a theoretical approach can be adopted, where we take into consideration the protein molar mass and the theoretical protein concentration in the food, including the natural variation of protein expression in the food ingredient. This can be achieved with matrix like milk or even egg white. Following this approach, the largest contributor to the expanded uncertainty of the measurement is the conversion factor. When the food ingredient is less characterized, the allergen abundance can be determined experimentally by relative quantification by discovery mass spectrometry. This approach relies on the availability of reference material for food allergen analysis, which are actually becoming available to the analytical community. It also strictly depends on the availability of full-length curated protein databases, which unfortunately are not always available for food allergenic ingredients. So, can we achieve comparable analytical measurements? Yes, we can, when the analytical method is well developed. The isotopic dilution mass spectrometry-based quantification of allergenic ingredients in chocolate, for example, provided comparable performances by 4 laboratories down to 4 ppm level of contamination for some of the marker peptides that were used as a reporter of allergenic food protein ingredients. Measurement scientists, risk assessors, food industry should work together to create a fit-for-purpose measurement infrastructure to support food allergen labeling. The optimization is a key criteria for isotopic dilution mass spectrometry methods to allow the accurate quantification based on the selection of representative allergenic peptide markers to report for total allergenic protein. And reference material for food allergen analysis, which are becoming available to the community, will help to support the harmonization of the measurements and also the generation of proper conversion factors that would help translating the peptide concentration to the total allergenic protein.